Hey class, in this presentation we are going to start talking about eukaryotic cells, particularly the organelles within a eukaryotic cell. So let's talk about eukaryotes. To review briefly, a eukaryote is a cell that has a nucleus, or a true kernel. It's believed that a eukaryotic cell appeared about 2 to 3 billion years ago, BYA. Bacteria, archaea, and eukaryotic cells are believed to have all evolved from the same common ancestor, or last common ancestor. The first very primitive eukaryotic cells have been believed to be very single-celled and independent. Multicellular eukaryotes are more recent in the geologic timeline. As time has progressed, eventually these single-celled eukaryotes would form colonies, and then these single-celled eukaryotes that are in colonies began to specialize. And this is the theorized progression from single-celled to multi-celled organism. These complex multicellular organisms started as individual cells and lost the ability to live individually, to live by themselves separate from that colony because of those specialized functions. As we look at our categories of multicellular or eukaryotic cells, we'll have our protozoa, algae, fungi, and helminths. Protozoa are always going to be single cellular or unicellular. Helminths, or parasitic worms, are always going to be multicellular. Excuse me. And then finally, the fungi and the algae go back and forth. Some are multicellular, some are single cellular. So, let's pause for a moment. Which of the following characteristics is evidence that mitochondria were once in prokaryotic cells? Well, we haven't answered this question yet. So let's come back to this after we're done talking about the organelles of the cell. Here is a diagram showing a simplified version of your classic eukaryotic cell, and we kind of threw everything, including the kitchen sink, at this diagram. It has lots and lots of organelles in it. Even though it's nice and round, it will have a nice flagella, and it'll have our chloroplast, our Golgi apparatus, our mitochondria, the centrioles, the nucleus, the endomembrane system, so on and so forth. So let's talk about those organelles. We'll first start off with Cilia and Flagella. The eukaryotic flagella, unlike the bacterial flagella, has some very distinct differences. It is about 10 times thicker, and it's much more complex, structurally speaking. It has an extension of the cell membrane that covers it. And within the center of it, there's a long cylinder that contains regularly spaced microtubules. Another thing to note about the eukaryotic flagella is that it moves with a whipping fashion compared to the bacterial flagella, which moves with a spinning, rotating fashion. The eukaryotic flagella has a primary function of moving the cell. Eukaryotic cilia, on the other hand, are primarily going to be used to move a substance past the cell. They are very similar to the flagella in structure, but are smaller and more numerous. They are found in only a single group of protozoa and certain animal cells. In human beings, we have cilia that will line our lungs, our respiratory tracts. We also have cilia that line our uterine tubes, aka fallopian tubes. And then in multicellular organisms, those cilia are used to move a substance past the surface of the cell. In that single group of protozoa that has cilia, this is the exception to the rule, that single group of protozoa uses cilia for locomotion. Around the surface of the cells, we have the glycocalyx. Just like with the bacterial and archaeal cells, the glycocalyx is the outermost layer that comes in direct contact with the environment. It has a lot of polysaccharides. It can sometimes appear as a lot of fibers that are interlinked with each other. Sometimes it's very slimy in appearance, or sometimes it's more waxy in appearance, forming a capsule. These glycocalyx allows for cells to adhere to each other, adhere to a substrate, and to process intercellular signals. The cell wall that is associated with our eukaryotes is not going to be present for every single eukaryote. For example, protozoa and helminths do not have cell walls. It also is worth noting that animals do not have cell walls, but plants and fungi do have cell walls, just like bacteria do have cell walls. Within fungi, the cell wall is going to not be made of peptidoglycan like it is with the bacteria, but instead the cell wall of fungi is made primarily of chitin. Chitin is what I want to emphasize to you. Now, the cell wall of the fungi is very rigid, just like with other cell walls. It gives the cells rigid structural support. It has a very thick layer of chitin that makes it up. Every now and again, you'll see a fungi with cellulose in its cell wall, and then external to that, there's going to be a thick layer of mixed glycans. Glycans are a very resistant category of molecule that resist abrasion and add additional strength and stability to the fungal cell wall. So here we can see some yeast, some Candidus albicans. And as we zoom in on the border of that cell, we'll have the cell membrane on the inside, and then here we have the extracellular environment over here. And we start with chitin, that's in direct contact with the cell membrane. Then we'll have some glycoproteins that link the chitin to the thick layer of mixed glycans. And then finally, the glycocalyx, or carbohydrate shell around that, will interact with the external environment. The cell membrane, sometimes referred to as the plasma membrane or cytoplasmic membrane, is a phospholipid bilayer that will have proteins embedded in it. Sometimes we'll also have steroid hormones embedded in it, cholesterol being the single most common steroid hormone that's embedded within the cell membrane. As we have a higher concentration of sterols, we'll have a more viscous membrane. Organisms that are adapted to live in higher temperatures have higher concentrations of sterols in their cell membrane than organisms that are adapted to live in lower temperatures. Also, organisms that do not have a cell wall, generally speaking, will have a higher concentration of sterols compared to organisms that do have a cell wall, because those sterols make the cell membrane or plasma membrane a little bit more resilient and more viscous, so it's not as easily distorted. This cell membrane also is going to be so selectively permeable. It lets some things go in and out, and other things it blocks and will not allow through. In the center of the cell, we have the nucleus. This is the namesake of the eukaryotic cell. The nucleus is easily visible under a light microscope, even if you use just a simple methylene blue or iodine tincture as your contrasting stain. The nucleus is distinct from the cytoplasm because it has a double membrane, a double phospholipid bilayer, known as the nuclear envelope. This nuclear envelope has two membranes with a very small space in between them, and to allow chemical information to go in and out of the nucleus, this double layer, this nuclear envelope, needs to have small holes or nuclear pores that will allow for the information to travel through the membrane. In the middle of the nucleus, we have a region known as the nucleolus. It is found within the nucleoplasm. The nucleoplasm is a term that refers to the liquid on the inside of the nucleus. Within the nucleolus, we'll have RNA synthesis. tRNA, rRNA are the two that are primarily synthesized in the nucleolus. This is an area for where we make the ribosomes of our eukaryotic cells. So you and I, as human beings, as eukaryotes, will have a nucleolus within our nucleus, and that is where we make the ribosomes of our cells. Chromatin is a term that I remember struggling with when I was younger, because I always taught DNA is in the nucleus, DNA is in the nucleus, and then some people started telling me that chromatin is in the nucleus. So what's the difference? Chromatin is a term that refers to both DNA and proteins collectively. It's a more generic term. DNA is a more specific term. Within the nucleus, we don't have the DNA of the cell floating around unorganized. It's wrapped around some proteins to help it keep organized. Those proteins that the DNA wraps around, much like thread being wrapped around a bobbin, are called histone proteins. The DNA, to be more specific, is the genetic material of the cell. The chromatin is the DNA wrapped around the histone proteins, or organized DNA. So here we can see scanning electron micrograph of a nucleus. In the center of the nucleus, we have the nucleolus. And then right next to the nucleus, you could probably be able to see a lot of squiggly lines. Those squiggly lines that are right next to the nucleus make up the endoplasmic reticulum. The nucleus is going to be continuous with our next organelle, the endoplasmic reticulum. They are merged with each other. So let's talk about our endoplasmic reticulum. It is a series of tunnels that connects directly to the nucleus that's used for transport and storage. And there are two endoplasmic reticulums, or ERs, that we're going to talk about. We have the rough endoplasmic reticulum, sometimes also referred to as the rough ER or the RER. Most people will say rough ER. This rough ER, or rough endoplasmic reticulum, is going to allow for materials to go from the nucleus to the cytoplasm to the cell's exterior. So if a protein is going to be excreted, or a compound is going to be excreted from the cell, it's probably going to start out at the rough endoplasmic reticulum. The rough endoplasmic reticulum gets its appearance because it is covered in itty-bitty tiny little ribosomes, or it has ribosomes giving it a polka-dotted or rough appearance. If we go back a slide here, you can see a bunch of little blue dots on this endoplasmic reticulum. Those blue dots represent ribosomes, so we would call this a rough ER. The smooth ER, unlike the rough ER, does not have ribosomes all over its surface. It is used for nutrient processing, so there will be detoxification of toxic chemicals, and there's also going to be lipid production and storage occurring within the smooth ER. Here's a better figure showing how the membranes right here on this electron micrograph, this big sandwich for pancake full of membranes, is right next to the nucleus. This is a very well developed endoplasmic reticulum in this cell. Each individual little ribosome that's on the surface of the endoplasmic reticulum is going to be making a protein, or synthesizing a protein, and that protein is going to be secreted into the inside of the rough ER. It's also worth noting that you can have multiple ribosomes right here. We have 3 ribosomes that are all secreting a protein, and they're all reading the same piece of genetic information. They're all reading the same piece of messenger RNA. After the ribosome, the next organelle that's typically covered is the Golgi apparatus. We make the proteins with the ribosomes at the rough ER. From there, the proteins will be sent to the Golgi apparatus, when they're modified, repackaged, and shipped out. The Golgi apparatus has a lot of flattened dish-shaped cells called cisternae. In many respects, the Golgi apparatus looks like another endoplasmic reticulum that's away from the nucleus. It's very, very closely associated with the endoplasmic reticulum, though. We have transitional vesicles from the endoplasmic reticulum that are picked up on the face side of the Golgi apparatus, and within the endoplasmic reticulum, the proteins that are carried there will be modified within the cisternae. Each cisternae is going to have a slightly different chemical environment. Those different chemical environments allow for chemical modification. Oftentimes there'll be complex carbs or lipids attached to the proteins during this modification process in the Golgi apparatus. And then finally we have condensing vesicles on the on the far nuclear side of the Golgi apparatus. These condensing vesicles will pinch off and then carry cells to lysosomes, or carry proteins to lysosomes, or carry proteins to the extracellular environment for secretion. So from the ER, we'll have a transitional or transport vesicle bud off and then merge with the Golgi apparatus, and then the proteins are modified, potentially having lipids or carbs added, and then condensing vesicles come out the other side. Here is a Golgi body in a photomicrograph. One thing to note is that we have a series of flattened stacks, and it is not next to the nucleus. If you see a series of flattened stacks right next to the nucleus, it's probably going to be an endoplasmic reticulum. If it's not next to that nucleus, think Golgi apparatus. You think of the nucleus to endoplasmic reticulum to Golgi apparatus as an assembly line, sometimes also referred to as the endomembrane system. We'll take a piece of genetic code, or DNA, from the nucleus, make it into messenger RNA, and that messenger RNA is going to go from the nucleus through the nuclear pores into this rough endoplasmic reticulum. From the rough endoplasmic reticulum, that piece of messenger RNA is going to be picked up by a ribosome on the surface of the rough ER, and will be used to synthesize a protein that's secreted into the inside, or the lumen, the hollow space on the inside of the rough ER. When you see this term lumen, lumen refers to the hollow space within a structure. And that protein will go from the lumen of the rough ER and be transported to the Golgi apparatus. From the Golgi apparatus, the protein is going to be modified, repackaged, and then used by the cell, probably for secretion or to be secreted or to be embedded within the cell membrane. So here's a slide summarizing this process. We go from the nucleus to the rough ER, and then from the rough ER to the Golgi apparatus, and then to secretion. And that's only if the ribosomes are located on the rough ER. There are other ribosomes in the cell. If the ribosome is in the cytoplasm of the cell and synthesizing a protein, that ribosome is going to be synthesizing a protein that's probably staying inside of the cell. If the protein synthesized at the rough ER, it's probably going to be secreted. The vesicles that come off the Golgi apparatus are sometimes referred to as lysosomes, sometimes vacuoles, depending on what their purpose is. A lysosome contains a wide variety of digestive enzymes. These lysosomes will serve to process large food molecules that are brought into the cell, and they are also going to process damaged organelles within the cell. From an extracellular perspective, lysosomes are used to help remove cellular debris and damaged tissue from multicellular organisms to help keep the organism running optimally. We also will store needed biomolecules within a eukaryotic cell, and the storage structure is known as a vacuole. A vacuole is a membrane-bound bag, or a sac, so to speak. You can think of it as essentially just a suitcase inside of the cell. Sometimes it stores chemicals that are going to be used at a later time within the cell. Sometimes it stores chemicals that are going to be secreted at a later time from the cells. Phagotic cells are known for having a high number of vacuoles, and it will form vacuoles in response to phagocytizing food or other substances that are being brought into the cells. After that food is brought into the cell within a vacuole, that vacuole will then merge with the lysosome, and that will digest the products within the vacuole. So here we can see a big chunk of food particle, and it's being brought into the cell. It's been phagocytized, and then the cell has a membrane wrapped around it, a vacuole wrapped around it. Now the Golgi apparatus starts sending lysosomes to that vacuole containing the food particle. The lysosomes merge and release their contents into the food vacuole, and then the food is digested. We also need energy to run our cells. The power plant of the cell is the mitochondria. It generates cellular energy, adenosine triphosphate ATP. We get most of our ATP from our mitochondria for eukaryotic cells. The mitochondria is a two-membrane organelle. There's a smooth outer layer and then an inner layer, or inner membrane, that folds back and forth on itself. The folds within that inner membrane are known as cristae. The cristae within the mitochondria hold many enzymes and electron carriers that are used for aerobic respiration, and these enzymes are used to extract the most possible chemical energy from the food that goes into the cell, primarily in the form of glucose, especially if we're looking at human cells, and uses that to make ATP. Mitochondria are a very unique organelle. They divide independently of the cell, and these mitochondria have their own DNA. There is mitochondrial DNA. Mitochondria also have their own ribosomes as well. The fact that mitochondria have their own genome and have their own ribosomes and divide independently of the cell has led many scientists to hypothesize that mitochondria used to be separate bacterial cells that used to live independently of the eukaryotic cell, but now have formed a symbiotic relationship with the eukaryotic cell. Here is a diagram showing our mitochondria. We have the smooth outer membrane and then the bendy inner membrane, and then there's a little bit of DNA and some simple ribosomes, 70 Svedberg ribosomes within the mitochondria. Another organelle that's common to many eukaryotic cells is a chloroplast. A chloroplast is found predominantly in cells that go through photosynthesis. So you'll see them in algal cells and within plant cells. The key thing of a chloroplast, or the key function of a chloroplast, is to take energy from the sun and turn it into chemical energy. So that chloroplast is going to make glucose. It will take our carbon dioxide and water and use those compounds to make organic carbon. A byproduct of photosynthesis is oxygen, and thankfully we as humans breathe in that oxygen. Something that's similar between the chloroplast and the mitochondria is that the chloroplast, just like the mitochondria, has a smooth outer membrane and then has an inner membrane with lots of folds in it. Chloroplasts are larger than mitochondria, though, and contain special pigments, notably chlorophyll as a special pigment, and have some more variety in the shape of their structures. The next organelle we're going to talk about is the smallest organelle. Most of the organelles we've talked about up to this point, every organelle I've talked about up to this point have been membrane-bound organelles. The ribosome is different. It is not a membrane-bound organelle, and it's distributed fairly evenly throughout the eukaryotic cell. Some are going to be free-floating in the cytoplasm, some are going to be attached to the cytoskeleton, some are going to be embedded in the membrane of the rough endoplasmic reticulum, and then there's also special ribosomes, or chloroplastic ribosomes, or mitochondrial ribosomes that are going to be within the lumen, or hollow space, inside of the mitochondria, or inside of the chloroplast. Our ribosomes are found in short chains, sometimes known as polyribosomes. These polyribosomes are capable of all synthesizing the same protein at the same time from the same piece of genetic information. Our ribosome has a large and small subunit. If you recall from our presentation on prokaryotic cells, that prokaryotic cells have smaller ribosomes at 70 Svedbergs. Eukaryotic ribosomes, generally speaking, are 80 Svedbergs in size. And it's a combination of a large subunit and a small subunit. I explained this in the other recording, but I just want to emphasize it again in case you didn't watch the other recording yet. Svedbergs are not a unit of mass. 40 plus 60 is not equal to 40 plus 60 in most situations is going to equal 100. So how do I get 80 Svedbergs out of this? Svedbergs don't add like when you add mass or when you add cups or when you add traditional units. A Svedberg is a special unit that measures how quickly something will move through a column of gel when it's being centrifuged. And since it's a centrifugal unit, things behave kind of weird or differently compared to units of mass and units of volume, which is what we primarily work with. What I want you to focus on is that eukaryotic ribosomes are bigger than prokaryotic ribosomes, and eukaryotic ribosomes have a large and small subunit. To give structure to our eukaryotic cell, we have the cytoskeleton. The cytoskeleton anchors the organelles within the eukaryotic cytoplasm, is used to move RNA and transport vesicles throughout the cytoplasm, and changes the shape of the cell. As the cells change shape, this can be used to facilitate the movement of the cell. Within the cytoskeleton, there are three primary elements. We have actin filaments in our eukaryotic cytoskeletons. We'll have microtubules, and then we'll have intermediate filaments. Our actin filaments are the thinnest structures. They are the smallest strands within the cytoskeletons. The microtubules are the largest structures within the cytoskeleton. They are long and hollow compared to very thin and narrow like the actin filaments. And then we have intermediate filaments, which are in between actin and microtubules. So here we have some strands of our cytoskeleton. In orange we have a thicker microtubule, then in pink we have a thinner intermediate filament, and then kind of in this grayish purple color we get in the background, you can see very thin wispy actin filaments. The cytoskeleton can be tagged so that it would fluoresce specific colors under a fluorescent microscope, and you can see the strands of the cytoskeleton in this photomicrograph in the lower left corner of your screen. Here is a summary slide summarizing the differences in between prokaryotic cells, so our bacterial archaeal cells, our eukaryotic cells, and something we haven't talked about yet, viruses. The general trend is eukaryotic cells are the most complicated, they're the largest and the most complicated. The second largest and most complicated are going to be the prokaryotic cells, and then the smallest and simplest will be viruses. So, concept check, guys. Which of the following organelles do we use to make energy in the cell? You can pause the video and look up the answer right now. 1, 2, 3, 4, 5. It is the mitochondria. And we had another concept check at the beginning. Oh, there we go. Which of the following characteristics is evidence that the mitochondria were once prokaryotic cells? So you can pause right here. So hopefully you looked up the answer by now. Our mitochondria have prokaryotic ribosomes. They have the 70 Svedberg ribosomes. They are capable of independent division. And what hasn't been mentioned in the PowerPoint, but through the process of elimination, is if we have two correct answers right here, then we must go with E. All of the above choices are correct, which means that mitochondria also have circular ribosomes and also have membranes that can be inhibited by antibiotics. If you have any questions about the material or the mitochondrial merger hypothesis, please feel free to shoot me an email or post it on the discussion board. Happy studies!